Hey, welcome to The Explainer. Today we're diving deep, like microscopic deep. We're going to figure out how scientists make sense of the totally chaotic, invisible world of bacteria. I mean, how do you even tell one from another? Let's get into it.
Think about it. It's a huge challenge, right? They're invisible. They're literally everywhere. And there are trillions and trillions of them. It's kind of like trying to map out a kingdom you can't even see. So where on earth do you start?
Well, it all kicked off back in the late 1800s. And believe it or not, it was with a surprisingly simple trick. One physician figured out a way to split the entire bacterial kingdom into two giant groups, and all he used were a few drops of dye. That's it. And that physician's name? Hans Christian Graham. But here's the craziest part. He wasn't even trying to classify bacteria. Now, he was just trying to make them easier to see under his microscope. And in this total aha moment, this happy accident, he stumbled upon a fundamental difference that pretty much changed microbiology forever.
Okay, so to do this, you need what's basically a four-part toolkit, and you have to use it in a really specific order. First, you've got your main color, the primary stain. Then, you've got this stuff called a mordant. Think of it like a primer or a glue that makes the color stick. After that, a decolorizer, which is basically an alcohol wash. And last, a counterstain to add a different color to anything that got washed out.
So, the whole procedure is a pretty straightforward four-step recipe. You stain it, you set it, you wash it, and you stain it again with a different color. Let's actually walk through what's happening on the slide.
Okay, step one. You flood the slide with this stuff called crystal violet. And boom, instantly everything, every single bacterium on there just soaks up this deep purple color. Right now, they all look exactly the same. No difference at all.
All right, next up is the iodine. This is that mordant we talked about, the glue. It gets inside the cells and basically grabs onto the purple dye, forming this big chunky molecule. And because it's so much bigger now, it's way, way harder to wash out. And here we go. This is the big one, the moment of truth. The alcohol wash. This is where everything changes. You pour the alcohol on, and for some of the bacteria, it just washes that purple dye right out. Poof, they're colorless. But the others, they hang on to that purple for dear life. And just like that, you have two different groups. So for the final step, we add a pink stain called safranin. Now, the purple guys are already full of color, so nothing happens to them. But those colorless bacteria, they soak up the pink. And there you go. Two totally distinct groups on one slide. The purple ones, we call those gram-positive, and the pink ones are gram-negative.
So you got to be asking, why the difference? Why do some hold the color and others don't? It's all about their cell walls, basically their armor. The gram-positive bacteria have this really thick, dense wall that traps that big purple dye molecule. But the gram-negative bacteria, their wall is much, much thinner. So the dye just washes right out. And this isn't just a fun party trick. That difference in armor is absolutely crucial for things like, figuring out which antibiotics will work against them.
Okay, so the gram stain is awesome. It splits them into two huge teams, Team Purple and Team Pink. But what if you have two different bacteria and they're both purple? You need another way to tell them apart. So the next thing scientists look at is, do they move? And if so, how?
A lot of bacteria have this amazing little tool to get around called a flagellum. And honestly, the best way to picture it is like a tiny little outboard motor. It's this long, whip-like tail that spins around and propels the bacterium through whatever liquid it's in. And I have to say, this is not just a simple tail. A flagellum is an incredible piece of natural engineering. It's a real, genuine, molecular motor. You've got the basal body, which is embedded in the cell wall and literally rotates like a wheel. Then there's a hook that works like a universal joint, and the long filament part is the propeller. It's just amazing.
So when it comes to classifying them, this gives us our first super simple yes or no question. Does it have a motor or not? The bacteria that don't have any flagella at all, we call those atrichous, and the ones that do, they're called trichous.
But okay. For the ones that do have flagella, it's not enough to just say yes. The arrangement of those flagella is the next big clue. So let's do a quick tour. First up, the simplest one, monotrichous. Mono means one, So it's just a single flagellum, a single propeller at one end.
Then it starts getting a little more complex. You've got Lophotrichus bacteria, which have a whole tuft of flagella all bunched together at one end, kind of like a powerful ponytail pushing them forward. Then there's Amphitrichus. Think of a boat with a motor on the front and the back. They've got a flagellum at each pole, so they can switch directions in an instant. And finally, you have Peritrichus. Peri means around, and that's exactly what this is. These guys have flagella sticking out all over their entire surface. This gives them amazing mobility, letting them kind of tumble and swim in any direction they want.
All right, so we've got color, we've got how they move. The third big piece of the puzzle, and maybe the most obvious one, is just what shape are they?
First up are the cocci. These are just the round, spherical ones. If you see just one, it's a monococcus. A pair is a diplococcus. And if they form a chain, that's streptococcus. And yep, that's the strep in strep throat.
The next main category is the bacilli. These are all the rod-shaped bacteria. And just like the cocci, how they're arranged is also a clue. You can find them as single rods, in pairs, or linked up in long chains. And to round out our little shape gallery, we have the curvy ones. You've got the long, corkscrew-like spiral shapes, and then the ones that look like little commas. Those are called vibrio. And hey, notice that callback to our last section? These twisty, curvy types are almost always moving, pushed along by their flagella.
So, let's just put it all together. To get that first basic idea on an unknown bug, scientists lean on these three pillars: how it reacts to the gram stain, whether it moves, and how its flagella are set up, and its basic shape. It's this methodical process of looking at clues that lets us take this invisible world and finally start putting names to faces, so to speak.
And all of this leads to the really big question. So what? Why does it matter if we know a bacterium is, say, a gram-negative Peritrichus bacillus? Well, it's not just trivia. Knowing that tells us so much about its basic biology, its defenses, and really importantly, its weaknesses. So if this is how we identify a bacterium, what does that knowledge let us do? What comes next in medicine or industry or just understanding life on this planet? That's the real game changer.